Hello, so my name's Andrew Bowman. I'm a group leader in Warwick Medical School. So I run a small research lab. We are mostly focused on looking at how DNA is packaged within the nucleus of a eukaryotic cell, and also substructure of chromosomes in interphase nuclei. So on the integrated science course, I'm going to be running the second block with my colleague Michael Smutney, and that is B2 in Codebase of the cell. So this module is aimed at giving you a basic introduction to molecular biology, but also giving you an introduction to a working lab environment and teaching you some basic molecular biology tools. So the seminar component of this module will be revolved around basic or an introduction to molecular biology, and we start off with central dogma, and this is how information flows in biology. So genetic information we know is encoded in DNA. So here. This information first has to be converted to a similar molecule called RNA, and this is done through a process known as transcription. And this can also be reversed, so you can get RNA to DNA. For example, certain retroviruses have RNA genomes, and they first convert them to DNA before they're integrated into the host genome, such as HIV. And then when the information is an RNA, this is converted to protein, and this process is known as translation. So first we look at this flow of information and how hereditary material is encoded. So you probably all know this from your A-level biology. But DNA consists of basically one of four bases. So you have adenine, thymine, guanosine, and cytosine. These pair specifically A with T and G with C into the double helix. But in terms of proteins, these are made up of amino acids, but you have 20 different protein subunits. So you have alanine, cytosine, aspartate, glutamate, phenylalanine, and so on. So we look at how DNA with just four bases can encode proteins with their 20 amino acids. In an example here, we show that it can't be a single base to an amino acid because that would be 4 to the power of 1 or 4. It can't be two bases because that would be 4 to the power of 2, 4 times 4, which would be 16. And in actual fact, it's three bases code for a single amino acid. So 4 to the power of 3, 4 times 4 times 4, which is 64. As you can see, 64 is much larger than 20. So we also look at the degeneracy of the genetic code. And we also look at how you could potentially improve the way that organisms encode genetic information. So we also look at gene structure in this module, as you can see here. Basically how genes are organized, where genes know where to start, once you have a specific triplet nucleotide for where they start and also where they stop. We look at directionality in the chemical structure of DNA. We say going from 5' to 3'. And we also look at how genes are regulated through their promoters so organisms can respond to their environment. We also look in detail how the process of transcription works, the enzymes involved. It's an enzyme called RNA polymerase is responsible for transcription here to convert the gene to RNA, and then how RNA is converted to protein, as you see here, the little Pac-Man. So we look at the ribosome enzymes that are involved in this process and how this can be regulated. And further, we look at protein structure, its primary, secondary, and tertiary and quaternary structure, and essentially how this provides function, and thereby linking the phenotype of the cell or the genes, the DNA sequence, genotype to the phenotype, i.e. What you actually see in reality in the environment. We also have a little look at genetic engineering, how this knowledge of how the information of life is encoded, how it's been used in the lab beneficially for therapeutic means, for example. So we look at how you can take a gene from humans, use specific enzymes to clip this gene out, and then stick it or ligate it into a vector or kind of a mini chromosomes from another organism, in this case bacteria, a much simpler organism, and then how you can propagate these simple organisms in the lab. And also how you can then express human genes in this bacteria to make particular proteins of interest. In our example here, where we have the human insulin gene, you can create a human insulin protein rather than purifying it from pigs, which I had to do in the past. So this example here is showing how understanding the basics of molecular biology has helped therapeutically in treating diabetes. And also, as I said, this is one component of this module is to introduce you to basic lab techniques. So the project we're actually going to work on in this module is the cloning and purifying of an engineered protease. So a protease is a protein which degrades other proteins. So here you can see a protease as a Pac-Man, and here is its target protein here, going from the N-terminal end to the C-terminal end. Protease basically chops this up, snips this up into its either small peptides or individual amino acids. So a number of proteases will degrade proteins just to amino acids. So, for example, there's a number of unspecific proteases in your digestive tract, like pepsin or trypsin, that will degrade protein that you ingest so that your gut can absorb it. Proteases are also prevalent in other areas of life, and we're actually going to look at a viral protease. So a number of viruses actually, instead of producing one gene, one protein, they actually produce or have a very long gene, and this produces a polyprotein. That's what you can see here. So this polyprotein has 1, 2, 3, 4, 5 different proteins encoded in it. And in order for these to be functional, they have to be released from this polyprotein. And here is where a protease comes into play. So in this example, we're going to, or in this lab, we're going to use a protease, which has found a lot of use in the biotechnology field, and that's a protease called tobacco etch virus protease, or TEV protease. This is from a virus that infects tobacco plants. And this protease basically recognizes a specific amino acid motif that you can see here. So glutamate, asparagine, leucine, tyrosine, phenylalanine, glutamine, serine, and it cleaves specifically between the glutamine and the serine there, the Q and the S. And this produces the individual proteins, the viral proteins, which then go on to create more viruses. So within the practical, we're basically going to synthesize some DNA, which will encode a certain string of amino acids, namely the recognition site for the TEV protease, and we're going to stick this into a bacterial vector which has a tag on one side to help in purification, and it has a reporter on the other side. So this reporter is GFP, or green fluorescent protein, which I'm sure you've probably heard of. And this green fluorescent protein acts as a reporter because it will fluoresce green under UV light, so it will tell us where this protein is. And essentially we're going to assess the kinetics of different engineered TEV polymerases, which have been recently published. We're going to test their kinetic activity by using this reporter construct that we clone in a digestion reaction over various time periods. So this is an example here showing how we separate out proteins, and this is a process done. This is done by a process called SDS Page. So the page stands for polyacrylamide gel electrophoresis. So essentially we'll make a gel, and then if you add your unfolded proteins to this gel, and you run them, or you expose them to an electrical current, they will actually migrate down the gel according to their size. So smaller ones will migrate faster, larger ones will migrate more slowly. So we're going to do digestions with our protein reporter constructs over, for example, 0, 1, 2, 3 minutes. And then what we should see is that the reporter construct migrates at the top, should disappear, and the GFP on its own, which is smaller and therefore migrates faster, will start to appear on the bottom. We'll look at different ways to quantify these gels and use those to measure the kinetics of the enzymes. So I hope that's given you a little insight into the module and what we aim to achieve. And as I can't take questions, I'll be happy to see you all in October.